Hello friends, welcome to my channel Pharmatech Competitive. So our today's lecture orientation is the nervous system. It is much more complex in structure and functions when compared with the other visceral organs and systems. It is also unique in having blood-brain barrier and in lacking lymphatics. The system comprises of the central nervous system or CNS and brain, and the peripheral nervous system, that is PNS, and spinal cord and peripheral nerves. Both these parts of a nervous system together acts as a command and control center of the body. Its derangements are fundamentally different in that they may organic or functional disorders are mental diseases which are due to the disturbed biochemical pathways or due to the impairment of the intersynaptic transmissions. Organic diseases are large number of diseases having abnormalities in the structure and function. However, here only common and important diseases having morphological changes will be discussed. That discussion is divided under two main headings, central and peripheral nervous system, each of which is preceded by the review of applied aspects of the normal structure. First one is central nervous system and second one is peripheral nervous system. We will study the normal structure and the diseases associated with it. So, do subscribe our channel for more such video lectures. So let us start our today's lecture. Let us discuss about the central nervous system normal structure. The skull and the vertebrae form a rigid compartment encasing the delicate brain and spinal cord. The average weight of the brain is about 1400 gram in men and 1250 gram in women. The two main divisions of the brain, the cerebrum and the cerebellum, are quite distinct in the nature. The brain does not have lymphatic drainage. There are two types of the tissues in the nervous system. First one is neuroectodermal tissues, which include neurons or nerve cells and the neuroglia, and together form a predominant constituent of the CNS. The mesodermal tissues are microglia, dura mater, and the leptomeninges, that is pia arachnoid, blood vessels and their accompanying mesenchymal cells. The predominant tissues comprising the nervous system and their general responses to the injury are briefly considered below. First one, neurons. The neurons are highly specialized cells for the body which are incapable of dividing after the first few weeks of the birth. Thus, brain damage involving the neurons is irreversible. Neurons vary considerably in size and shape. Their size may range from the small granular cells of the cerebellum to large Benz cells of the motor cortex. Some neurons are found, others oval or fusiform, but phototype of the cortical neurons is pyramidal in shape. A neuron consists of three main parts, that is cell body, an axon, and numerous dendrites. Myelin sheath envelops the axon that can be demonstrated by the special stains such as Luxol fast blue. The cell body is a main constituent of the neuron from which an axon and numerous dendritic processes extended. The cell bodies may be arranged in the layers in the cerebral cortex or may be arranged as in the basal ganglia. The cell body possesses a large round centrally placed nucleus having finely granular nuclear chromatin and a predominant nucleus. The cytoplasm contains polygonal basophilic structures called Nissl substances. It consists of aggregates of RNA, sheaves of rough endoplasmic reticulum, and the intervening groups of free ribosomes. Besides Nissl substance, other special features of the cytoplasm of the neuronal cell body are presence of microtubules, synaptic vesicles, and neurofilaments, which are a form of intermediate filaments specific to neurons. Lipofuscin may be present due to aging. Neuromelanin is found in neurons in the substantia nigra and pigmented nucleus of the pons. Neurons respond to injury in a variety of ways depending upon the etiological agent and the pathogenic processes. These include central chromatolysis, atrophy, and the degeneration of the neurons and axons. The intraneuronal storage of substances. Neurophil is the term used for the fibrillar network formed by the processes of all the neuronal cells. Second, neuroglia. The neuroglia provides supportive matrix and the maintenance of the neurons. It includes three types of cells: astrocytes, oligodendrocytes, and ependymal cells. Neuroglia is generally referred to as glia. The tumors originating from it are termed as gliomas, while reactive proliferation of the astrocytes is called as gliosis. Astrocytes. The astrocytes are stellate cells with numerous fine branching processes. In a routine hematoxylin and eosin stains, as astrocyte has round and oval vesicular nucleus, but unlike neurons, lacks a predominant nucleolus. The cytoplasm is generally scanty. The processes radiate from the cell body. Depending upon the type of processes, two types of astrocytes are distinguished. A. Protoplasmic astrocytes have branches of processes and are found mostly in the gray matter. And B. Fibrous astrocytes, they have long, thin processes and are present mainly in the white matter. Some astrocytic processes are directed towards the neuron and their processes, while others surround the capillaries by the terminal expansions called as foot processes. The astrocytic processes may not be visible by the routine stains but can be demonstrated by the phosphotungstic acid hematoxylin, that is PTAH stain. Ultrastructurally, these processes are composed of abundant intermediate filaments, mostly vimentin, that can be demonstrated by the glial fibrillary acidic protein, that is GFAP immunostain. Oligodendrocytes. Oligodendrocytes are so named because of their short and fewer processes when examined by the light microscopy with special stains. In hematoxylin eosin stained sections, these cells appear as small as cells with a darkly staining nucleus resembling that of the small lymphocyte. The cytoplasm appears as clear halo around the nucleus. Oligodendrocytes are present throughout the brain in gray as well as white matter and are most numerous of all the other cells in CNS. In gray matter, they are clustered around the neurons and are called satellite cells. In white matter, they are present along with the myelinated nerve fibers and are termed as interfascicular oligodendroglia. The major function of oligodendrocytes is the formation and maintenance of the myelin. Thus, in this respect, they are counterparts of Schwann cells of the peripheral nervous system. Diseases of the oligodendrocytes are therefore disorders of myelin and myelination, such as inherited leukodystrophies and acquired demyelinating diseases. Ependymal cells. The ependymal cells are epithelium-like and form a single layer of the cell lining the ventricular system, adequate central canal of the spinal cord and cover the choroid plexus. They are cuboidal and columnar cells and have ciliated luminar surface, just beneath which are present small bodies termed as blepharoplasts. Ependymal cells influence the formation of the composition of the cerebrospinal fluid by the process of active secretion, diffusion, absorption, and exchange. The function of cilia is not very clear, but probably they play a role in circulation of CSF. The ependymal cells respond to injury by cell loss, and the space left is filled by the proliferation of the underlying glial fibers. Microglia. Microglia is the nervous system counterpart of the monocyte macrophage system. Although the term microglia is commonly used, but it is inappropriate since these cells, unlike neuroglia, are not neuroectodermal origin. Microglial cells are not fixed but are mobile cells. These cells are found throughout the brain and are often present close to the blood vessels. Normally, microglial cells appear as small inconspicuous cells with bean-shaped vesicular nuclei, scanty cytoplasm, and long cytoplasmic process in the response to injury or damage. However, these cells have capability to enlarge in size, proliferate, and develop elongated nuclei, so-called rod cells. Microglial cells may actually assume the shape and phagocytic functions of macrophage and form glitter cells. The foci of the necrosis and areas of selective hypoxic damage to the neurons are surrounded by the microglial cells, which perform phagocytosis of the damaged and necrosed cells. This is known as neuronophagia. Dura mater is the tough, fibrous covering of the brain, which is closely attached to the skull on its inner layer of the endocranial periosteum in the region of the spinal canal. It encloses the potential space, the epidural space between the bone and the dura. The dura is composed of dense collagen fused with periosteum of the skull. Pia arachnoid or leptomeninges. The leptomeninges, where lepto means thin or slender, considering of the pia and arachnoid matter, form a delicate vesicular membranous covering of the central nervous system. The pia mater is closely applied to the brain and its convolutions, while the arachnoid matter lies between the pia mater and the dura mater without dipping into the sulci. Thus, a space is left between the two layers of leptomeninges known as subarachnoid space, which contains the CSF. The major arteries and veins run in the subarachnoid space, and small nutrient arteries pass to the cortex. Extensions of the subarachnoid space between the wall of blood vessels entering the brain and their pile sheets from a circumvascular space called Virchow-Robin space. Another important potential space is enclosed between the dura and the arachnoid membrane is known as subdural space. Some facts about the normal structure of CNS you must know. First one, there are two types of the tissues in the nervous system: neuroectodermal tissues and mesoectodermal tissues. Neuroglia is the supportive matrix of the neurons and includes astrocytes, oligodendrocytes, and ependymal cells. Astrocytes are of two main types: protoplasmic and fibrous. So let us move towards the questions that can be asked based on this topic in the semester examination. Question is, describe the normal structure of the central nervous system for 10 marks and describe the structure of neuron with the well-labeled diagram. So the answer should include all the points as we discussed. Next question that can be asked based on this topic is, there are two types of tissues in nervous system, that is A. neuroectodermal tissues, B. mesodermal tissues, C. both A and B, and D. none. So the correct answer is option C, that is both A and B. As there are two types of tissues in the nervous system, that is neuroectodermal tissues, that is neurons or nerve cells and neuroglia, and mesodermal tissues, that is microglia, duromater and leptomeninges or pia arachnoid and blood vessels are found. So here we conclude our lecture. In the next